Hello, everyone, and welcome to today's webinar. My name is Michelle Woodhill, and I'm the Chief Development Officer at Dr. Susan Love Foundation for Breast Cancer Research. For those of you who are joining us for the first time, I'd like to take an opportunity to introduce you to our organization. Dr. Susan Love Foundation challenges the status quo to end breast cancer through innovative research and improve the lives of people impacted through education and advocacy. We drive collaborative cutting-edge research with non-traditional partners, shed light on the collateral damage of treatment, and actively seek ways to diminish it, and interpret science to empower breast cancer patients. Many of the projects the foundation undertakes are designed to increase our understanding of the human breast, where breast cancer starts and what enables malignancy to occur, with the ultimate goal of developing methods to prevent it in the first place. The foundation is also committed to participation in research that has the potential to positively impact the lives of people who are at risk for or who have breast cancer. Breast cancer happens in people, so research needs to be done on human beings, and that means men, women, non-binary people, everyone who gets breast cancer. Our Love Research Army is committed to encouraging and facilitating breast cancer research in all people, regardless of age, race, gender, sexual identity, with or without breast cancer diagnosis. We are committed to thoughtfully engaging our members by providing them with ongoing real-time access to an array of studies. Today's webinar is part of our webinar education series, where we focus on topics of interest to our patient community. Thank you to our supporters of this program, Daiichi Sankyo, Seattle Genetics, and Merck. Joining us today are Dr. Sarah A. Hurvitz, Professor of Medicine at UCLA, Dr. Sarah Horton, Executive Director of Access and Diversity at Quantum Leap Healthcare Collaborative, and Susan Colum, Program Director of BreastCancerTrials.org. Our panelists will be available to answer specific questions related to their presentations from our viewers at the end of the webinar. So please submit your questions using the Q&A box at the bottom of your screen. While we will try to answer as many questions as possible, please note that we may not be able to address every question today. Our first presenter is Dr. Sarah A. Hurvitz. Dr. Hurvitz is a professor of medicine at the University of California, Los Angeles, co-director of the Santa Monica UCLA Outpatient Practice, medical director of the clinical research unit of the Johnson Comprehensive Cancer Center at UCLA, and director of breast oncology. Dr. Hurvitz earned her MD from the University of Southern California. She served as internship residency at UCLA and was chief resident of internal medicine and completed a hematology oncology fellowship at UCLA in 2006. Dr. Hurvitz received board certification in internal medicine, hematology, and medical oncology. Thank you so much for joining us today, Dr. Hurvitz.
Thank you so much for having me. It's my pleasure today to talk about clinical trials. I'm going to do sort of a sweeping review of what clinical research is and sort of focusing on cancer clinical research. Next slide, please. So this slide is really trying to make the point or demonstrate the difference between clinical research on the right and basic research on the left. Basic research is research that we think of occurring in the context of a laboratory or bench research. This is the first time that a theory or a hypothesis is tested. It's original. It's easy to reproduce because a lot of times the work being done at the bench in the laboratory is being done on cell lines or small animal models. So you can do the experiment over and over and produce the same results. And it's a relatively efficient use of time and resources. It's fairly inexpensive, and you can get answers pretty quickly in the course of days, weeks, or even months. Clinical research, once we move drugs into human beings, it's now what's called clinical research. And this is ensuring that the new intervention being tested is both safe and effective, and it's getting the product ready for marketing and commercialization to human beings. It's important that these studies be conducted in human beings to make sure that they are safe. These studies allow the development of new knowledge to optimize our medical practice, but they're very expensive to conduct. There's a lot of ethical regulations when you're conducting research on human beings, as there should be. And so these are longer studies. They're often years to complete. And as I mentioned, very expensive. And it's a challenge to repeatedly do the same experiment over and over on human beings, as you could imagine, that just adds to the time and expense and may not be an ethically correct thing to do. Next slide. Now, translational research is a term that was coined sometime in the 1990s, which is meant to represent the interface between the bench, the laboratory, and clinical research. It used to be the laboratory would test early hypotheses. A drug, for example, would get ready to go into the clinic. It'd be tested in the clinic and then get approved or not approved. But now there's much more give and take, where the hypotheses from the laboratory lead to clinical trials, but the clinical trials, we are now very interested in obtaining blood samples and tumor samples and other information that can go back to the laboratory so that we can better understand biomarkers that help identify patients who have responsive disease or resistant disease. So it's a much more give and take process, and translational research sort of is the term that's coined to represent that give and take. Next slide. So what is a clinical trial? It's a research study to answer specific questions about new medical interventions. I'm talking a lot today about drugs, but this also could be a device or lifestyle factors, a new diet, for example, that are aimed to prevent or screen or diagnose or treat a condition. Clinical trials are used to determine whether these interventions on humans are both safe and effective, and carefully conducted trials are the fastest and safest way to find new interventions that work in people. Next slide. So here are some questions that could be addressed by clinical trials. There are prevention studies. For example, large studies were done to see whether or not taking a drug called tamoxifen can prevent breast cancer in women who are at very high risk. There are trials that can be done to look at whether doing imaging can pick up cancer early, and if that imaging picks it up early, does that improve outcomes? Those are screening trials, lots and lots of mammography screening trials. And then the focus of a lot of research is therapeutic clinical trials, which are interventions done to treat a particular problem, such as breast cancer. And then there are studies done to make sure that there aren't any harms from this intervention. And most of our trials will touch on both therapeutic benefits as well as safety. Next slide. There are a number of phases of clinical trials. There are 4 phases. Actually, there's 5 because there are some studies called phase 0, but for the purpose of our discussion, phase 1 is a first-in-human study where the drug is first given to human beings. Sometimes there are like 5 or 6 different phase 1 trials of a particular agent tested in different types of diseases. These are usually very small studies. They're opened only in a few centers and very, very close oversight to make sure this patients are safe and to measure the effects of the drug. Phase 2 are larger trials after the phase 1's been successfully completed. Often it's one-arm studies, so everybody gets the same treatment, although there are some where you have a couple of different arms. And then there are phase 3 trials, which are the trials that are conducted to get a drug or intervention approved by the FDA. It's the final step before getting FDA approval, and these are very large studies. Often there are several different groups that, at least 2 groups that patients can be assigned to, and you compare the outcomes of the 2 different groups, and if it's a positive study, it gets approved. Phase 4 is after a drug has been approved, and these are called post-marketing studies, and they're done to evaluate after a drug's been approved, whether or not that drug has efficacy or effectiveness in a real world population, and to ensure that the safety and efficacy reflects what was shown in the phase 3 trial. Now, all of these clinical trials are different from observational studies. Observational studies are done where you don't do anything. You just look at a group of patients and look at what's happening. For example, example, smokers versus non-smokers. You're not giving them cigarettes, but you're monitoring the rates of cancer in patients who are smoking versus people who are not smoking. That's an observational study where you're not intervening. Clinical trials, there's always an intervention. Next slide. Next, just go ahead and hit the button. So only 1 out of 50 agents tested in the laboratory are going to actually make it to phase 1 testing. The chance of success of a phase 1 study is about 49%. That means it'll move on to phase 2. And then the success rate of phase 2 trials getting to phase 3 is only 1 in 4, 25%, and only about a 50-50 chance that a phase 3 trial will be positive. So if you think of that, it's a very low probability that cancer drugs from the laboratory are going to make it through FDA approval. And a typical new drug application to the FDA in the 90s consisted of 68 clinical trials for one drug, 4,200 patients, and that included several phase 3 studies, a number of phase 1 or 2 studies. Next slide. And actually, recent data shows the likelihood of approval based on the disease type being studied for an intervention. So, for example, at the top, you can see 24% of hematology drugs go from phase 1 all the way through phase 3 and successfully are approved. Oncology is near the bottom. Only 5.3% of drugs that make it to phase 1 that are cancer drugs make it all the way through to phase 3 and get approved. So you can see the success rate is quite low. Next slide. Now, phase 3 controlled clinical trial is depicted here. And in phase 3 trials, a group of participants who meet eligibility criteria and are enrolled in the study are randomly assigned to either receive an experimental treatment or a standard treatment. And the randomization has to be by chance. It has to be random. It can't be that the investigator decides who goes in which arm, and it can't be that the patients choose which arm they want to be treated in, because that could insert bias into the clinical trial where patients might be sicker in one arm of the study or not. And the patients are followed in each arm for the outcome. For example, are they alive at the end of the study, or are they not alive? Or has their disease returned, or has it not returned? And you compare the differences in the rates of those events between those two treatment arms. Next slide. So why do we randomize patients? Why don't we allow patients to decide which treatment arm they'll go in, or why can't the investigator choose that? And as I mentioned, we have to evenly distribute factors that could confound our results, both measured and unmeasured factors. And confounding factors are features of the patients that could affect the outcome being measured. For example, breast cancer recurrence or survival. If sicker patients are in the experimental arm, they may not live as long, not because the drug wasn't working, but because there were sicker patients in the treatment arm. So we want it to be by chance or random, and want to make sure that any effect that's measured in the trial is due to the intervention, the drug that's being tested, not due to an imbalance in the treatment arms. This avoids assignment bias, where the investigator influences which patients get which treatment, I've already gone over this example, so go to the next slide, please. So why do we blind participants or investigators to treatment? If a patient or investigator knows which arm the patient's been assigned to, it can bias the interpretation. And so what we do is we blind the investigators and the patients, and if you blind both, it's called double blind, to what treatment arm they've been assigned, so that any measurement, for example, has the disease grown or not, is unbiased by knowledge of which treatment arm the patient was getting. Now, sometimes studies have to be done in an open label or unblinded fashion. And in that case, you can blind the radiologist to what the treatment was that the patient was getting. So the radiologist who's making the measurement and call whether the disease is getting worse or whether it's shrunk is doing it in an unbiased fashion. Next slide. And then a word about placebo. Placebo is not used generally in phase 1 studies and usually not in phase 2 studies, but in phase 3 studies, it may be used to help blind patients. And it is expensive to do placebo-controlled clinical trials because you need staff that are unblinded and know which treatment the patient is getting. And it's important to know that almost never is the placebo the only treatment being given in a cancer trial. Usually all the patients are getting an active study drug, and it's just that you don't know if they're getting something additional or if they're getting which study drug they're getting. So on the next slide, you can see, next slide, that a placebo-controlled trial example where patients were assigned to receive anastrozole plus a placebo. Everybody's getting anastrozole, an active drug, or a placebo that looks like anastrozole and a new oral drug. And the next slide is another example. And this is a study where patients, and this is actually a controlled trial, anastrozole was given with a drug called palbociclib or with a placebo. So nobody knew if they were getting the palbociclib or placebo, but everybody was getting anastrozole. So I think placebo scares a lot of prospective clinical trial patients thinking they're not going to get an active treatment, but modern ethically run studies do not omit therapy for patients with cancer. Next slide. So I'm going to leave this for Dr. Horton to discuss, which is the importance of having a diverse population that represents the population who will be receiving the treatment once it's approved, because it's a critical issue in trials. Next slide. I'm going to skip this one, and this one, because in the interest of time. So what these slides are about is essentially endpoints. Every trial has to define what are you measuring to see if the drug works or not. The gold standard endpoint in cancer trials is how long a patient lives, but patients live a very long time sometimes with cancer. So we need to use earlier endpoints such as, does the disease slow down their growth? Is there shrinkage of the disease? Does the drug improve the amount of time that patients can live without the disease getting worse? And these are called surrogate endpoints that help the trials be conducted in a more efficient manner, both financially and temporally. Next slide. So my last couple slides is the journey for the prospective trial patients. And a patient who is a clinical trial begins by an investigator having a written protocol or a recipe book saying exactly how the clinical trial would be conducted, who's going to be eligible, what happens to a patient on the study, what is the rationale for the study, how will we manage toxicity, what are the endpoints, what are the statistics to say whether or not the study was positive. And once that protocol is written and informed consent, the investigator obtains IRB or ethics board approval. The investigator then, once they have approval, can present the study to a prospective patient. If the patient's interested, after reading the consent in full, the patient can sign the informed consent with the investigator in person. Typically, it's important to know the consent is not a contract. The patient can withdraw their consent at any time for any reason. It's more of a permission slip to participate in this procedure or this experiment. Next slide. Screening for the study is usually 21 to 28 days, during which time the investigator and their team make sure the patient meets the eligibility criteria for the study. Once they're found to be eligible, they enroll in the trial. And if it's it's more than one arm of the study, they'll be randomized to one of the arms. The study treatment then starts following the protocol very strictly, and the length of study participation for metastatic breast cancer usually is as long as the disease is responding and the drug is well tolerated. But in early stage cancer, they usually define a period of treatment and follow-up. Next slide. And oversight is key. There have been serious wrongs done in the past in the name of clinical research. Tuskegee Airmen is just one name that I think rings a bell for many. But there are now many ways that oversight is ensured to make sure that studies are done ethically based on good scientific rationale. And this is overseen by the FDA, who's involved in approving all studies, evaluating new unapproved drugs or new combinations. The study sponsor who's making the drug has their own monitors. There's data safety monitoring boards, IRBs, radiation safety committees, scientific peer review committees. And in fact, with all of this oversight, there is emerging data that patients who do participate in clinical trials for cancer may have better long-term outcomes. And it may be not due to the intervention being studied, but due to the very strict oversight and standardization of patients' care. Next slide. And with that, I'll close. I think I'm a couple minutes over. I apologize. Thank you so much.
Thank you so much, Dr. Hurvitz, and no problem. Next, I'd like to present Dr. Sarah Horton. Dr. Horton is a board-certified medical oncologist who currently serves as the Executive Director of Access and Diversity for Quantum Leap Healthcare, a nonprofit organization that focuses on the support of breast cancer clinical trials. She also holds an affiliate associate professorship at Howard University Cancer Center and serves as a medical oncologist for the Howard University Cancer Center Transdisciplinary Breast Cancer Clinic. Dr. Horton has dedicated her career to improving disparities in cancer outcomes and increasing diversity in clinical trials. She has over 20 years of oncology experience in academia, clinical practice, regulatory medicine at the FDA, and advocacy. She has held several leadership roles at Howard University Hospital, previously serving as their Chief of Hematology Oncology, Director of Cancer Clinical Trials, and Associate Program Director of Hematology Oncology Fellowship Program. She has published multiple papers on disparities in cancer outcomes in African American patients. At the FDA, she served as a medical officer reviewing clinical trials for breast cancer therapies. And during that time, she created a 6-week FDA Oncology Summer Scholarship Internship Program for minority and underrepresented high school students and co-founded the Oncology Diversity Working Group. Dr. Horton completed her undergraduate degree at Northwestern University, medical degree at Wright State University School of Medicine, and her Hematology Oncology Fellowship at Georgetown University Lombardi Cancer Center. Thank you so much for joining us today, Dr. Horton. And I'd like to introduce you to Sarah.
Oh, thank you, Michelle. Thank you for that introduction. So we have heard a lot of great information from Dr. Hurvitz. So thank you for that on clinical trials. And like she alluded to, I will be focusing on the importance of diversity in clinical trials today. So representation is important. We hear that phrase a lot. It's especially important when it comes to science, though, in terms of our understanding about how things work. So I'll start today with some numbers to help give a better understanding of where we are in terms of diverse representation in clinical trials. So to put things in perspective, statistics show that today about 49% of the US population is made up of racial or ethnic minorities. When you look at the clinical trials that were performed to approve the drugs in the US, 2 to 16% at a high are comprised of the ethnic or racial minorities. When you look at African Americans, we make up 14% of the US population, and about 5% or less are represented on trials. With Latinos or Hispanic patients, they're now making up 18% of the US population, and again, represent usually 1 to 5% of clinical trial participation. So along with racial and ethnic, Along with racial and ethnic minorities, there are several different types of groups that are underrepresented on clinical trials. Today, I'm just going to focus on the racial and ethnic minorities, and that usually consists of Black or African Americans, Hispanic or Latino patients, Asian Americans, Native Americans, Alaskan Native, and Pacific Islanders. Next slide, please. So how do we know that racial and ethnic diversity in clinical trials is low? So several studies have been performed that are looking at the racial makeup of the clinical trials, especially in the US. In 2018, a paper was published in Nature that looked at the clinical trials that were used in the medications that were approved by the FDA between 1997 and 2014. So this is over a 17-year period. What it showed was that over the course of those years, there wasn't much of a change in the racial makeup of the patients involved. One group that we did see a small increase in is the Asian population, where we went from 0.25% in 97 to 6% participation in 2014. But when you look at the rest of the populations, it stayed just about the same, the minority being non-Hispanic white patients. Next slide, please. So why is representation important? We know that therapies may differ in their safety and efficacy based on the patient. So race, ethnicity, gender, sex, they all impact how a patient may respond to the same treatment. So when you don't have diverse patients in your studies, you run the risk of making assumptions about how that drug is going to work in the body that may not be accurate. Studies with diverse participants ensure that your therapies and your prevention efforts have a greater efficacy and safety across all populations. Next slide, please. So barriers. When we look at reasons why we don't have good diverse participation in clinical trials, you can look at it from different perspectives, from the clinician's perspectives in terms of barriers, from the patient, as well as from those who are developing the clinical trials, such as pharmaceutical companies and sponsors. Today, I'm going to just look at the clinician barriers and some of the patient barriers that we see more commonly. And from a clinician or provider standpoint, we know that sometimes lack of time, just being too busy, having too much work to do, having to see too many patients will deter a provider from talking about clinical studies. There might be provider bias, implicit bias, we call it, where they may feel that for some reason that patient may not participate in the study, be it a language barrier or just a ethnic barrier. And just insufficient cultural competency. The provider, and by provider, I mean be it the doctor or the nurse practitioner or whomever is in charge of talking about the clinical trial, does not feel that they can comfortably talk about studies, which can be complicated, to the patient in a way that they can understand. And this also includes gender identity barriers from providers. So another reason that clinicians may not speak to patients about studies is some health centers are under-resourced. They don't have the funds to support a clinical trial program, so the trials are not available, or a few trials are available, and so it's just not discussed. And unfortunately, people of color are more likely to receive their care at these places. A lack of knowledge of the studies in the area. There's some providers who are just not interested in research, and because of that, don't provide it as an option to their patients. And then the lack of workforce diversity, meaning the lack of diversity in the providers, can be a barrier to patients. It may increase the chance of having provider bias, and as well as not necessarily being sufficient with cultural competency to talk to the patients, to recruit them and retain them on the studies. Next slide, please. In terms of the patient barriers to clinical trials, patients may not be informed. So I put this under patient barriers, which would basically be a patient not knowing about studies. But actually, the onus lies in the providers to tell them about the studies. And there's been a research poll in 2017 that showed that 24% of Black patients stated that their doctor or practitioner ever even spoke about medical research. We know that language barriers on the patient's side may be a deterrent, not understanding the study or feeling that they may not understand the complexities of the studies, may be a barrier for them to participate. There still is some well-earned mistrust in the community about the health system, some of it historic, some of it present day, in terms of mistreatment of Black and people of color, as well as the LGBTQ community. Comorbidities or other medical problems that a patient may have, such as diabetes, heart disease, hypertension, may exclude patients from clinical trials. And again, that has to do with eligibility criteria. The people who write the studies put that criteria in. And these comorbidities tend to occur at a higher rate in communities of color. Financial challenges where the patients may not have insurance, and so they wouldn't have ways of covering the treatments. There are study-related travel expenses, childcare expenses, if they have to come in multiple times for appointments, and time off from work to participate. Next slide, please. So given these problems with the low representation of diversity in clinical trials, the first steps we took were to try to look at where we are today in terms of diversity in clinical trials. And this was undertaken by the FDA. So this slide from the FDA is looking at cancer drug approvals over a 10-year period. And what you see from this slide quickly is that over 10 years, when you looked at the racial makeup of the patients in the clinical trials that were used to approve those drugs, it remained pretty consistent that there's around an 80% participation of non-Hispanic white patients. And if you look at the bottom of the graph, you'll see both the Hispanic and Black population tends to make up less than 10% across the board over the 10-year course. Next slide, please. Because of this, and to provide more transparency in terms of the type of patients involved in the clinical trials, that were being submitted to the FDA for the approval of the drugs, the FDA started a new initiative called the FDA Drug Trial Snapshot Program. Their goal was to share information on the diversity of the patients in the clinical trials and publish this in a summary report annually. That way, anyone who was interested in the type of patients that were used to approve a drug could go to the FDA website and look up that drug and find out exactly the demographic makeup. Next slide, please. So what we found in 2021, and there were 15 oncology drugs approved in 2021, on the snapshot was that there remains a racial imbalance. So there were about 75% or higher participation were white in the studies, less than 5% were Black, and less than 6% were Hispanic. When you look at the 2021 general US population Census Bureau numbers, 64% of our US population is white, 17% is Hispanic, and 12% is Black. So it just kind of highlights that we have a lot of room to improve to get close to reflecting what the makeup of the US population is. Next slide, please. So when offered to participate, when thinking about some of the reasons minorities were not participating in clinical trials, often we heard that providers felt that minority patients would not participate in a clinical trial if asked, or were suspicious of research. And so because of this, a meta-analysis or a paper was published in 2021 looking at a series of studies that were particularly done, and there were 35 in total, that were looking at clinical trial participation rates. And the studies that they looked at had patients by different races, and they were asked, would you participate in the study? And so in the yellow box is kind of the takeaway points for me. This is looking at numbers compared to white patients. So if you look in the column of Black patients, in the yellow box, there were 13 studies, and 58% of the Black patients said yes when asked, compared to, in the same study, 55% of white patients. And you'll see this going through the Hispanic and Asian populations as well. In their studies, 6% of the Hispanic patients would have agreed to participate in the same study that 61% of whites were asked, and same with Asians. And so that study, when this article was published, it pretty much confirmed what we were suspecting and knew, that if asked, people of all races would more likely participate in a study than anyone else. Next slide, please. So to understand why we feel that participation is important, we have to understand that we have differences, real differences, in how well or how poorly different racial groups do with different diseases. And we call this a health disparity. And an example of a health disparity is with breast cancer in Black women. That's the example that I'll use today. So you may or may not have heard these numbers, these statistics about Black women when they're diagnosed with breast cancer, that Black women are at a 42% higher chance of dying that compared to white women, there's a 39% higher chance that the breast cancer will come back once treated compared to white women, and a 71% higher relative risk of death in general if given the breast cancer diagnosis compared to white women. Right. One of the possibilities to explain some of the Black-white disparity could be that the drugs may not work the same in different populations. So with our capabilities of identifying molecular and genetic changes or biomarkers in patients, it may be that you have a higher percentage of a certain type of biomarker in a racial group, and that may affect how the drug works. But if that racial group is not reflected in the clinical trial, we'll never know that that's the case. We'll never know what that genetic change or biomarker the genetic makeup of the patients are, if they harbor that biomarker, and how the drug works in these specific bodies if they're not represented in the studies. Next slide, please. So that's the hard truth about clinical research. And this was so eloquently said by Dr. Jonathan Jackson at Mass General, who's the founder of Community Access, where he said inadequate minority representation in clinical trials means that we aren't doing good science. If we aren't doing good science and releasing drugs into the public, then we are at best being inefficient, at worst being irresponsible. Next slide, please. So we must do better in order to address the worst outcomes or the disparities that we're seeing and the inequities in cancer healthcare. We must bring all underrepresented populations into cancer research. Next slide, please. And now that we know how important it is to be involved in clinical trials, the next question is, how do we find clinical trials? And to speak more about that will be my colleague at Quantum Leap Healthcare, Susan Colin. Thank you.
Wonderful. Thank you so much, Dr. Horton. And as Dr. Horton mentioned, our last presenter is Susan Colin. Susan is the program director of breastcancertrials.org, a program of Quantum Leap Healthcare Collaborative. She's devoted her career to providing clinical trial education and personalized trial matching for patients. Since 2009, Susan has helped build BCT's portfolio of products and services, including metastatic trial search and metastatic trial talk. Susan considers herself a breast cancer survivor, having lost her mother to breast cancer when she was 5 years old, which drives her passion and commitment to advancing research and eradicating breast cancer. She received a BS in Industrial Engineering and Operations Research at the University of Massachusetts Amherst, and is a graduate of the UC Berkeley Extension Clinical Research Conduct and Management Program, as well as an MBCC Project Lead graduate and a member of the Metastatic Breast Cancer Alliance. Thank you so much, Susan.
Thanks. Thank you, Michelle. And thank you, Dr. Hurvitz and Dr. Horton, for that excellent information and the perfect segue. Now that you may be interested in clinical trials, how do you find them? So how do people typically find clinical trials? The most common way is by going to clinicaltrials.gov and the National Cancer Institute's cancer.gov, which are government-sponsored registries that list all of the clinical trials that are open and available in the US. Both of these sites are very comprehensive, but they were not designed for patients. The information is presented in medical and technical language, and the search feature can be confusing and hard to navigate, which is why and how breastcancertrials.org began. The idea for breastcancertrials.org, or BCT, came about in the late 1990s by two breast cancer patients who were interested in trials for themselves. They both had similar experiences where their doctors didn't tell them about trials, or they didn't know about trials other than those at their own hospital or institution. So they attempted to look for trials on their own. Both women were highly accomplished and educated women, but they both found it very challenging, frustrating, and confusing to look for trials on their own. One of the women did eventually find a trial by calling hospitals individually, one by one, and asking about their available trials. After both women completed treatment, they joined forces and came to the UC San Francisco Breast Care Center with an idea to develop a website that would make it easier for patients to find trials on their own. After 10 years of development at UCSF, with the participation of patients and oncologists. BreastCancerTrials.org launched as a nonprofit, free nationwide service in 2008. Our mission then and still is to empower all people affected by breast cancer to consider clinical trials as an option for their care. Next slide. We now have 3 programs that fulfill that mission, all related to clinical trial education and clinical trial matching. BreastCancerTrials.org is our original program. It's now used by people with DCIS or stage 1 to 3 breast cancer, as well as for breast cancer survivors, people who have completed their treatment for early stage disease, who might be looking for trials to reduce their risk of recurrence or manage side effects. Metastatic Trial Search is our companion application that was developed in 2015 as a collaboration with 5 breast cancer advocacy groups, with the idea of embedding it on their websites so that people can learn about and find trials on the trusted websites where they're already comfortable and familiar. And today you can find Metastatic Trial Search on over 35 partner websites, including on the Dr. Susan Love Research Foundation's website. Our third program is Metastatic Trial Talk, which is a monthly online newsletter designed to help keep people updated on what's new in metastatic breast cancer research and to learn more about the clinical trial process. We also list all of the new trials that were added to Metastatic Trial Search every month. Okay, next slide, please. So how are BreastCancerTrials.org and Metastatic Trial Search different from other matching tools out there? We've been matching patients to trials for over 14 years, and we're patient-friendly, meaning you can use these tools on your own to learn about and find trials. We provide the trial information in plain language. So we take the information that's available on clinicaltrials.gov and we rewrite the trial summaries in patient-friendly language to make it easier to understand. And we're also breast cancer specific, which allows us to focus entirely on breast cancer trials. We have a team who manually codes the relevant inclusion and exclusion criteria for each trial so we can match the right trials to each patient's specific breast cancer profile. And that's how we're really able to provide very accurate and useful trial matches. We're continuously improving based on feedback we get from our users. And lastly, we're a nonprofit service, which means that we don't sell or share patient data for money or any other purpose. And we don't accept money from drug companies to highlight their specific trials, which allows us to provide people with a comprehensive list of matched trials in an unbiased and equitable way. Okay, so I will now do a quick live demo for using Metastatic Trial Search to find trials. Oops. Okay, so you can use both of our matching services either as a guest or you can register and create an account. And there are benefits to registering and having an account, which I'll point out as I go. And for the sake of the demo, I'll continue as a guest, and you can always log in or register from any page going forward. So as a guest, we ask 6 questions related to your breast cancer diagnosis and demographics. You might want to have your medical records or your pathology report available. But you can, if you don't know an answer, you can always leave it blank or select unsure. But it's important to remember that the more information that you can enter, the more precise and accurate your trial results will be. So for the demo, I will select a hormone positive, HER2 negative patient profile. And I'm going to go quickly through these questions for the sake of time, but you can always contact us at any time if you have questions going through this questionnaire. So I will, for the demo, select liver metastases and a diagnosis date of February 2022. All of these questions that we ask are asked only for the purpose of determining eligibility for clinical trials. And this is a question about demographics, and which are optional, like all of the questions before. But what's important here is to enter your zip code, because the trials will then be sorted based on the distance from the zip code that you enter. And so I'm just going to point out here, these are 6 questions that you get asked as a guest user. But if you create a profile on our website, you have the opportunity for entering information about your treatment history. And many trials' eligibility criteria is based on whether or not you've had treatment before, and if you have, how many treatments and what kind of treatment. So by entering treatment history, you'll be able to get a more refined list. And as a guest on that profile we just filled out, we still matched to 367 trials. That's a lot. And I think we started at 501 trials. So by entering treatment history, you'll get immediately a more refined and accurate list. But there are other ways to filter the list as a guest user. And as you can see, we start by showing all of the trials across the US. You can firstly filter based on the distance from your zip code. So you can look at trials that are maybe only 100 miles from your location. And that will definitely narrow the choices, and it takes a minute or so, or a couple seconds, but we'll go from 367 trials for this profile to 37 trials in within 100 miles. There's other ways to filter trials also. One is by trial type. And we categorize these trials based on treatment trials or non-treatment trials. Non-treatment trials look at managing side effects and improving quality of life, amongst other things. And we also list surveys, interviews, and registries, which is a way for anybody to contribute to research. And most of these can be done by at home. So for the demo, let's say we wanted to look specifically at vaccines and immunotherapies. Again, for this profile. You can also filter by mutations and see trials that target these specific mutations if you have that information. Okay, so by selecting vaccines and immunotherapy, we will go from 37 trials to 10 trials. And you might, at this point, consider removing the radius, and you would be able to see immunotherapy trials across the US in case you are able to travel, or you might have family in other parts of the country. Okay, so now we're ready to look at trial information. And as I said earlier, we rewrite this information in lay language, including the trial titles. We also provide the official title or the scientific title, and this is the information you would see on clinicaltrials.gov. And it's available for people who might be interested in looking at the official titles. We do a high-level summary of the purpose or rationale, and we do extract all of this information from clinicaltrials.gov and just attempt to make it easier to understand. So we'll also provide the distance, the nearest location from your zip code, and a high-level summary of the burden or how many visits would be required if you participated. So what do you do if you find a trial that you are interested in? We always recommend that you share the information with your doctor, and you can share directly from our site by sending an email, and that email will have a link to the listing, and you can customize the message here. You can download a PDF, and you can copy the link and text or email the information to your provider or anybody else on your own. And you can also contact the research site directly if you have any questions. And we provide information, location, and contact information for the nearest site, as well as all the other sites where the trial might be conducted. And the information is provided based on, again, the clinicaltrials.gov data that we have. And finally, you can save the trial as a favorite. And this is a feature that is only available if you're a registered user and you're logged in on the website. And then you can view your saved trials through this tab at any time. So, you know, the benefits, again, for logging in or creating a profile is to be able to fill out a full profile, including your treatment history, saving trials as favorites, and returning to the site to see new trials that have been added in the last 2 weeks that matched your profile without having to re-enter your information on the site. And you can also easily update your profile with any changes to medication or any changes to your profile and do a rematch easily. So I just want to point out that we are looking at metastatic trial search, which is our matching tool for people with metastatic, living with metastatic disease. Breastcancertrials.org is available for people with early stage breast cancer. It will look and act a little differently than this, but we're actually working on converting that website to an application formatted just similarly to metastatic trial search for patients with early stage breast cancer. And the reasons we're doing that is so that we can embed a tool for early stage breast cancer on other websites, and that as metastatic trial search is, it will be mobile responsive and easy to use on mobile devices. So again, if you have any questions using our tool, you can always call us or email us at any time, and we're happy to help you walk through the website. And I think I have one more slide that has the links and the URLs to our tools, and hopefully left enough time for some questions. So thank you.
Thank you so much, Susan. Those are some fantastic resources for patients. And yes, we do have a little bit of time for questions. So I'll bring up some questions, and they're open to all of three of you. So whoever has any answers or feedback, I welcome them. We spoke a lot about increasing diversity representation in research. And so one participant asked, how can we reduce patient barriers and increase patient diversity in clinical studies and trials?
Okay, I'll take that. There's been a lot of work on what specifically can be done, like granular things that not only sites can do, but patients can do. One is, outreach and education, just like Susan's presentation on breastcancertrials.org. We're trying to make sure that if the provider doesn't speak about clinical trials, at least the community knows what a clinical trial is and that it's available. We're also, from a site standpoint, meaning the places where the clinical trials are being done, are focusing on what we call the social determinants of health, meaning things about someone's life that may make it difficult for them to be in a trial. So it could be transportation, right? So if a patient says, you know what, it's really hard for me to get here in the middle of the city to this site, weekly for 6 weeks, then sites are setting up Uber relationships where we can help with transportation. There are more and more translators who are on site to not only explain informed consent, but also to discuss the trial with patients. So there's definitely a focus on making it more inclusive with providers or even extended providers, be a patient navigator who can communicate with the patient, and then outreaching to the community, like churches and patient advocacy groups, places where patients trust the healthcare information that's coming out, possibly more than they do the institutions, to educate and about clinical trials, and then set up relationships so that this is something that that we hope will be ongoing and improving and stay for a long time.
Wonderful. Thank you, Dr. Horton. This question might be more directed towards Dr. Hurvitz, but for lower risk patients, what's the advantage for participating in a clinical trial?
Yeah, low risk is sort of a relative term. It's hard to address this question without specifics, but there are important clinical trials that are being done to see if less therapy is just effective as more therapy in patients who have lower risk breast cancer, for example. There are studies that are conducted to address whether you can do a shorter course of chemotherapy or omit chemotherapy. A lot of our clinical trials, I think, traditionally have been related to adding more therapy to higher risk patients, but there is a definite move toward what's called de-escalation, or reducing the amount of treatment as we become more sophisticated in identifying patients who have a lower likelihood of having a breast cancer recurrence. So there certainly are very good clinical trial opportunities that are addressing questions relating to whether less is good enough.
Mm-hmm. Thank you. Yeah, I have actually seen some of those studies, and it's pretty interesting. We have time for perhaps one more question. So let's see. What is your take on the I-SPY 2 endocrine therapy pilot study? And this person is wondering why there aren't regular updates on this trial in general. That might be too specific for this group to answer. So let's ask, could you please explain quote unquote hazard ratio and how that is reported in clinical trial results?
Yeah, the hazard ratio is often used. And the way without getting into all the specifics, it's basically telling you how much better or worse an intervention is than the control arm. So it's used when you're comparing two treatment arms, or you're comparing a new treatment to historical outcomes. But usually it's used in the context of a study that has multiple treatment arms. And when you're comparing the standard arm to the new treatment, you generate a hazard ratio. If the hazard ratio, what I do is I start with 1.00 and I select the hazard ratio. Let's say the hazard ratio is 0.70. So 1 minus 0.7 is 0.3, or 30%. So there's a 30% reduction in the risk of breast cancer coming back, or of death. So that's roughly how you do it. If the hazard ratio is higher than 1.00, then you have a higher risk of it occurring. So it's a statistical measure of how two arms compare, or two treatments compare to one another. And if you think of it in terms of a percentage, it kind of helps you understand the relative benefit or risk reduction of one arm versus the other.
Great. Thank you so much, Dr. Hurvitz. Well, we are unfortunately out of time today. I want to thank our panelists, Dr. Hurvitz, Dr. Horton, and Susan Colin, for participating today. It was incredibly informative, and I really appreciate you sharing all of the multitude of resources out there for our patient community. If there were any questions that weren't answered, please email us at info@drsusanloveresearch.org, and we will pass it on to the appropriate panelists to answer. And again, Susan shared the resources with breastcancertrials.org. Please utilize that service. I know Dr. Hurvitz answered a question. Clinicaltrials.gov is also another resource, but breastcancertrials.org does a nice way of filtering only specific to breast cancer trials. So thank you all very much. We truly appreciate your participation today. Thank you for everybody watching and listening at home, and we look forward to seeing you all at our next webinar. Enjoy the rest of your week.